A pharmaceutical company submitted an NDA after 3 years of development. Their clinical data was solid. 6 months later, they received an FDA complete response letter, not because the drug failed, but because a single leachable compound in a stability sample had no toxicological risk assessment behind it. 18 months and a complete container closure redesign. Today, we walk through 5 warning signs that appeared long before the later. Hello, I'm Raghuvar Singh Burma, Director Scientific Affairs at Resolve Mass Laboratories, and we work with pharma and biotech teams to identify and close ENL gaps before they reach a reviewer. Today, 5 warning signs your drug product is at risk of failing at the Extractables and Leachables review, and what to do when you find one. Why ENL reviews are getting harder to pass? So the regulatory bar for extractables and leachables has moved significantly in the last decade. PQRI laid the foundation in 1999, but the landscape shifted again with the ICH Q3E in 2023, raising expectations on toxicological risk assessment, analytical method design, and AET calculation. At the same time, FDA deficiency letters are increasingly citing ENL gaps in NDA and ANDA submissions, particularly for inhaled parenteral and ophthalmic drug products where the route puts compounds directly into the bloodstream or lung. ENL programs designed 5 years ago may not meet today's expectations, and teams that find out during review face the longest delays. Warning sign 1. Your container closure program was qualified early in the development and the formulation has changed since then. This is more common than most teams acknowledge. Formulations evolve, pH gets optimized, and a co-solvent is added, an excipient concentration shifts. Each change alters what migrates out of your primary packaging. The simulant solutions, temperatures, and time points in your original study were designed for the product that no longer exists. ICH Q3E is direct. Extraction conditions must reflect the final formulation. If you can't draw a straight line from your extractable study to the product you are filing, you have a gap. Warning sign 2. Your analytical methods were designed around USP sensitivity defaults, not the analytical evaluation threshold calculated for your specific product. The AET is derived from a toxicological risk assessment. It defines the lowest concentrations of a leachable compound that needs to be reported. If your method can't detect at that threshold, you are missing reportable compounds and the reviewer will find the gap. Multi-detector platforms, GCMS, LCMS, ICP-MS are not a nice to have for high-risk routes. They are the expectations and must have. Method validation must demonstrate sensitivity at AET level, not just a generic LOQ. Warning sign 3 comes in 3 forms, and any one triggers a deficiency request. Temperature. If your drug product is labeled for storage at 2 to 8 degrees Celsius, a study run at 40 degrees Celsius doesn't represent the patient's actual exposure. Regulators increasingly ask for the real-time data at the labeled conditions. Duration. A leachable study must run to the proposed self-life endpoint. A 6-month study for a 24-month label product leaves an obvious gap. Completeness. Every product contact surface must be included. Stoppers, plungers, tubings, adhesives, all. One untested component invalidates the entire package. Warning sign 4 is a change control problem. ICH-Q3E and FDA container closure guidance are clear. Any significant change to a primary container component requires ENL reassessment, a new supplier for your stopper, a resin reformulation, even a specification revision that looks minor on paper. The risk is highest when the supplier changes happen without direct notification to the drug manufacturer. At Resolve Mass, we have supported sponsors through packaging changes that began as a routine procurement decision and ended with a full ENL restudy and revised submission timeline. The gap almost always lives in a change control SOP that was never updated to flag ENL testing results. Warning sign 5 is a process failure. The correct workflow is TRA first, then AET calculation, then method design, then extractables, and then leachables. When teams reverse this and rationalize findings afterward, they discovered too late their method wasn't built to target what matters. Something most resources skips. The TRA is not the last step, it is the first step. 3 cases: a parenteral emulsion, an untargeted cyclic siloxane above SAT found after phase 3, 2-year delay. An MDI program, a valve supplier change not recognized as an ENL trigger until the CRL received. A biosimilar injectable accelerated stability data used for a refrigerated product, 14 months waiting for a real-time results. The good news, all 5 warning signs are identifiable before you file. Confirm your container closure qualification still reflects the final formulation. Pull the AET calculation and verify your method sensitivity reaches it. Audit your leachable study conditions against the product label, temperature, duration, and component coverage. Review change control records for packaging changes not flagged for ENL reassessment. And check when your TRA was completed relative to when your studies were designed. This 5-step check takes a morning. A complete response letter takes 12 to 18 months to resolve. So 4 things to take away from today's discussion. CCS qualification must match the final formulation, not the early phase placeholder. Calculate the AET before designing the analytical methods, not after. Every primary container component change is an ENL trigger. Build that into change control, and a prospective TRA prevents the reactive firefighting that costs submissions a year or more. In our coming videos, we dig into how to design an ENL study that passes FDA review on the first submission. If you are working on an IND, an ANDA, or a biologics program and need a CRO partner who treats extractables and leachables as a scientific discipline, not just a tick box exercise, Resolve Mass Laboratories supports small and mid-sized pharma teams from container closure qualification all the way through submission. Subscribe this channel so you don't miss what's coming and we can create more videos. Drop a comment on which topics you want us to cover the video, and thank you so much for watching this session until now. Goodbye for now. See you in the next one. Until then, goodbye for now.